Okay, so we're going to get started with the industry updates. So you guys heard from me just earlier today. So I'm Andy Berglund, Director of the RNA Institute, and I'm going to give you a little brief overview of the mechanism of DM1 and 2, and then we'll hear from our drug developer partners. So if I could have the next slide. Oh, never mind, I can control this. All right. So just to sort of set the field, we have our pharmaceutical and biotech industry folks. We have our patients, you and the families, the caregivers. We have the researchers, the clinicians, the therapists, the funding agencies. We just heard from FDA, the policymakers. And you're going to hear today from these 4 companies, and tomorrow there'll be 5 more companies. And just to give you an overview of the disease mechanisms of myotonic dystrophy. So you guys are all familiar with the CTG and the CCTG repeats, right? Many of you even know the exact number. So that's an area that, of course, you think about. And then what happens is the DNA is transcribed into RNA. And so that's that first green arrow. And that RNA is unfortunately the toxic RNA. So for DM1 and DM2, you have toxic CUG or toxic CCUG. And then what happens, and you saw this on a few slides already, is we have these RNA foci, and it's the toxic RNA in the cells. And what happens after that is that the MBNL proteins go down. Basically, you're losing the MBNL proteins, and the self proteins go up. And then an area that we don't have as much discussion right now, but is an important area, is something called RAN translation. And so the definitions are down at the bottom there. And then you hear a lot about the splicing abnormalities or the splicing changes. So that's sort of the next step. And then we have the clinical manifestations. And what's really exciting about all our companies is that, you know, they're shooting in all sorts of spots here. So it's not like we're just have the drug companies looking at just one area. So here are the 9 companies we're going to be hearing from today and tomorrow. And I just want to highlight Regenta, for example, tomorrow is hitting pretty far upstream, and they have a really exciting technology you'll hear about. We have 4 companies, Avidity, Dyne, PepGen, and Sanofi, all targeting that toxic RNA. And that's a really exciting area as well. And then you'll hear from Arthrex today, and they have a really impressive technology for upregulating the MBNL proteins. AMO Pharma has an approach that targets the self proteins. And then you're going to hear from Harmony and Juvena about their clinical approaches for hitting the clinical manifestations, so their therapeutic approaches. And I think one of the things that I'm most excited about is sort of all these approaches are complementary. And the way I think about it, and just to sort of give you the background, is we have the whole spectrum. So you're going to hear, well, most of our industry partners are beyond discovery, but there are people still in that space, and that's an important space to continue to develop the therapeutic approaches. Then you have target validation, you have lead optimization, process chemistry, preclinical development, phase 1, phase 2, and you'll hear about sort of all these aspects from the talks. And then phase 3 clinical trial. And then, of course, the FDA application and approval that you heard about earlier. So I think the things to remember when you hear the various talks is that our partners are taking a broad range of approaches to tackle DM. It's important to remember that the approaches span a broad spectrum of the pipeline. And yes. And so what I want you to also take away is that, you know, you're going to hear mostly about DM1, but a lot of these approaches are directly relevant to DM2. So that those of you with DM2 realize that the work has direct relevance for you. And some of those therapeutic approaches you'll hear about Arthrex and Juvena today and Regenta tomorrow. Those approaches that they're taking are directly relevant. For the ones that are more focused on DM1, the lessons that they're learning from their DM1 approaches can be directly translated to DM2 programs. And I know some of those are being developed. So I just wanted to highlight that for the DM2 folks out there. And then just to again highlight, engage and update your registry profile and find the opportunities for you and your family members, because not every one of these trials is going to be the right fit. So you with your clinicians are going to have to think about that. And then something that, you know, I love soccer. And the way I think about this now is it's a great time because we don't just have one shot on goal. We have lots of shots on goal. There's all sorts of sort of approaches, and we drew these soccer balls so that they're not all going to, say, the upper right corner. All the different approaches are hitting different places on goal. And hopefully within a short time, a few years, we're going to have a couple of shots that actually go in goal, and we're going to have FDA-approved drugs. So I think it's an exciting time to be in the field. All right, an exciting time for you. So we're going to hear from our partners that I'll introduce, and then these are tomorrow's partners. All right. There we go.
Hi, everyone. Thank you so much to the MDF for the opportunity to speak today. And I also want to say it's an honor for me to represent the Avidity team, to be here to tell you about the work we've been doing. But first, I really want to congratulate all of you for your involvement yesterday at the Hill. And it was so inspiring to watch. And also, I just want to commend the MDF team for pulling that off. It was not easy. So it's congratulations. So I will be making forward-looking statements, and the entire statement can be found on our company website. Let me spend this one slide to tell you a little bit about Avidity Biosciences. We are a small biotech company. Although our company size is growing, we now have 200 people. When I joined 3 years ago, there were only 50. We are located in La Jolla in San Diego, California, and patients is at the heart of everything we do at Avidity. Our mission is to improve the lives of people impacted by diseases with limited therapeutic options, such as myotonic dystrophy, through development of new class of drugs. As Dr. Johnson and Berglund said, DM1 is caused by the mutation that leads to an accumulation of toxic DMPK mRNA. So Avidity's strategy is to figure out a way to decrease that toxic RNA. The way we're doing it is through this product that we have developed from bench side now to bedside in the clinic. It's called AOC1001. AOC stands for antibody oligonucleotide conjugate. And the way it works is shown on this slide. So we can think of AOC1001 as composed of two parts, the blue part, which is an antibody, and the red part, which is an oligonucleotide, or oligo for short. The antibody is a class of molecule that can have high specificity to target another molecule. So we leverage this ability to bring the drug to the muscle cell surface and get internalized. And once internalized, the oligonucleotide is released, and it can go into the nucleus and also specifically bind to the DMPK mRNA and causes degradation. Today, I'd like to share with you some of the clinical data we have accumulated from our first in patient trial, which we call MARINA. It's a phase 1/2 trial. But before that, I really want to echo the importance of the work that you were doing yesterday. Governmental funding and support is crucial for basic science research. And basic science research is the only way we can understand disease biology. And it's only through understanding of disease biology that we can then develop therapies to treat the disease. So, and everything we're doing at Avidity has really been based on a lot of important work that's been made over the last several decades by advocates, experts, and we have been generating in this field knowledge and tools that allow us to then develop our drug. And some of these tools and some of these very important work is really listed here, including the Myotonic Dystrophy Clinical Research Network, the NDM1 Natural History Study, the patient advocacy and organizations such as the MDF, and also the community of patients, families, and caregivers such as yourself. And before I start, I really want to also, representing the Avidity team, to thank each participant, their families, the investigators, and their teams for their time, commitment, and continued contribution in the MARINA open label extension study. This slide, I'd like to use this slide to tell you a little bit about the MARINA trial. The purpose of the trial, there are actually 4 points. First one is to understand the safety and tolerability of AOC 1001. Second is to understand how the body process and eliminate AOC 1001. Third is to understand how AOC 1001 might affect the DMPK mRNA and the downstream molecules. And fourth, we also had exploratory endpoints to understand the clinical effect that we might see with this relatively short trial. So this trial is a 6-month trial. It's composed of 2 parts, part A and B. In part A, participants get 1 dose at 1 milligram per kilogram of AOC 1001, and then they're monitored for 6 months. In part B, there are 2 higher doses, 2 milligram and 4 milligram. In these cohorts, participants get 3 doses of either the AOC 1001 or placebo, and then they're monitored for a total of 6 months also. Eligible patients after finishing MARINA can enroll into the MARINA open label extension. And currently, all eligible patients have enrolled into the MARINA open label extension and are continuing to get treatment. This is a summary of the data from the MARINA trial. So first, AOC 1001 demonstrate improvement in myotonia and muscle strength and show early signs of improvement in mobility. AOC 1001 was observed to have a generally favorable safety and tolerability profile, with most common AE to be procedural pain and anemia. And most of the AEs were mild and moderate in severity. We did have 1 serious adverse event that was thought to be related to AOC 1001 in the 4 milligram per kilogram cohort. And this has resulted in a partial clinical hold by the FDA. This event is a very rare type of stroke that happened on both sides of the brain in a region called lateral geniculate nuclei in the thalamus. We have been working with the FDA over the last year to try to resolve the clinical hold, and we're continuing to work toward that. Based on the biopsy results from the MARINA trial, we know that AOC 1001 can be detected in the skeletal muscle. So we have successful delivery into the muscle, and it knocks down the DMPK mRNA and also positively impact the splicing change in the downstream of the DMPK mRNA. So just like what you have described by the panelists in the last session, we understand that hand function is a very important part of the disease. So I'd like to share with you this data. So in this is the data of from the assessment of we call video hand opening time, or VHOT. So the way it's done is by asking the participant to squeeze their hand very tightly for 3 seconds, and then to release as quickly and fully as they can. And this is recorded on a video and then rated by 2 blinded raters. And the duration it takes for the hand to open is summarized on the right-hand side of the slide. Let me walk through the format of the graph, because you'll see that in the subsequent slide. So the x-axis shows the study day in the trial. So day 183 would be 6 months in the trial. The 3 red arrows indicate the dosing date. And they're basically 6 weeks apart in the first 3 months. And then there's a 3-month of follow-up before the end of the trial. The teal color line shows the 2 milligram, the average summary result of the 2 milligram per kilogram cohort. The gray line is for the placebo, and the purple line is for 4 milligram per kilogram. So the y-axis indicate change from baseline. So if they had a decrease in the time it takes to open the hand fully, then there's an improvement and the line will go down. So as you can see, most strikingly, the purple line showing the 4 milligram per kilogram cohort had a 3-second decrease on average of the hand opening time after a single dose, and that this improvement is sustained through 6 months in the trial. This is an example of of a patient in the 4 milligram per kilogram cohort. On the left-hand side is the hand opening time video before any drug treatment. On the right-hand side is the days 183 or 6-month video hand opening recording after 3 doses of the drug. And these 2 videos are synchronized so that the word open are aligned. So you can see them being commanded to open at the same time.
Stay there.
As you can see, there's a pretty dramatic improvement that's detectable 3 months following the 3rd dose at 4 milligram per kilogram treatment. So what I wanted to actually add to the hand opening time is that the hand opening time is for it to happen, it needs the relaxation of the muscles in the inside of the hand, as well as contraction of the muscle on the outside of the hand. So we believe it's really a reflection of the hand function that happens in patients with myotonic dystrophy. So in addition to the strength that we were detecting through the hand opening time assessment, we also quantitatively assessed the strength in other muscle groups, and they are listed here. So we have hand grip, elbow extension and flexion, knee extension and flexion, and ankle dorsiflexion. On the right-hand side, we have the similar layout of the graph that shows the summary result of the 2 and 4 milligram cohort groups. And you can see that in this case, the stronger the patient has compared to baseline, the higher they should go on that graph. And this graph shows a dose-dependent improvement in the strength of the quantitative muscle testing result from the 2 and 4 milligram cohort. This is the last data slide I want to share today. So AOC 1001 also show early signs of improvement in mobility measures. And on the left-hand side, we have 10-meter walk run test for the duration for someone to do that. On the left-hand side, we have a time up and go, which is having a patient going from sitting in the chair, getting up, walk 3 meters, turn around, and then come back to the chair and sitting down. So that duration is summarized in the graph. So for both of these graphs, in a decrease in the duration means an improvement. And you can see that for the 4 milligram per kilogram cohort, again, we're seeing a trend toward improvement, even as short as 6-month treatment period. So in summary, our top-line data from MARINA reinforce our belief in the potential for AOC 1001 to be an effective treatment for people living with the devastating impact of DM1. We believe robust data package from MARINA supports a discussion with regulatory agencies about the design of a pivotal study. For the MARINA open label extension, it's currently ongoing, and all of the participants continue to stay in the trial and receive AOC 1001. And we have been in our discussions with FDA. FDA has been easing the partial clinical hold and recently allow us to increase the number of participants who can receive the 4 milligram per kilogram dose. And we plan to have a first look at data from this trial in the 2024, in the first half of 2024. So we remain steadfast in our commitment to advance meaningful therapy for the DM1 community, and we want to reinforce our gratitude for the study participants, families, advocacy partners, as well as the study investigators and their teams. And we are committed to partner with all of you to continue getting you information as we have them, and also resource as soon as they're available. I'll end with this slide. This is our Avidity family, and we are committed to continue to work on improving the lives of families living with myotonic dystrophy. And you can connect with us at the email listed on this slide. Thank you very much.
Hello, my name is Jeremy O'Connell, and I'm here representing the team at Juvena Therapeutics. I'd like to start by thanking the organizers and you, my fellow conference attendees, for giving me the chance to tell you about the work we are doing at Juvena to develop a drug for myotonic dystrophy. Some of the statements in this talk are forward-looking and should be interpreted in that context. At Juvena, we're passionate about developing new therapeutics by unlocking the therapeutic potential of stem cell secreted proteins. We do this with a machine learning enhanced biologics drug discovery platform we call Juvena that combines large experimental data sets with machine learning to help match the therapeutic potential in stem cell secreted proteins to particular indications. In executing on this pipeline, on this platform, we've filled a pipeline of promising therapeutics. And today, I intend to tell you about one particular candidate emerging from that pipeline for myotonic dystrophy, particularly type 1, but as noted by Dr. Berglund, we have a broader therapeutic focus than that and hope to speak in the future about other areas in which we are advancing therapies. In particular, I want to tell you about a molecule called JUV161. Show some of the data that give us conviction for its potential translation into myotonic dystrophy, and share some of our hope for entering the clinic in 2024. Given the great intro I had, I will actually be jumping over this and the following slides rather quickly. I just want to set up one particular point, as noted by Dr. Berglund and eloquently by Dr. Johnson before him, about the key changes in how cells respond when facing the stressors driven by the genetic mutation that produces the toxic RNA, and how cells respond to that, particularly how muscle cells respond to that, with changes in their ability to execute on their core program of surviving, differentiating, and forming new muscle fibers, and taking up and utilizing energy to produce functional muscle contraction and relaxation properties that present, again, clinically, as Dr. Johnson outlined more eloquently than I could here. In particular, I wanted to introduce this molecule called JUV161, which combines a portion of a stem cell secreted protein with a carrier protein called human serum albumin. And by combining these two, we create a natural, entirely protein-based molecule we're developing for subcutaneous administration with broad tissue availability to drive muscle strength and function and survival in patients. One half of the molecule is, as I mentioned, a natural carrier protein. It's the most common protein in the blood. And the other half is a stem cell or a portion of a stem cell secreted protein called IGF-2, whose primary function is in the earliest stages of embryogenesis for muscle formation, where it's one of the key drivers of the stem cells to divide and become new muscles. But the receptors and the processes for this molecule's action still exist in adults, and it still has potential efficacy. When we test this molecule in a mouse model of DM1, after 3 weeks of treatment, we see that there's no change in body weight in total, but we do see an increase in muscle mass, and importantly, an improvement in energy homeostasis, here measured by blood glucose levels. More importantly, though, we see that repeat treatment not only prevents the loss of muscle strength, but begins to show an improvement in muscle function by the 3rd week, here measured by forelimb grip strength in the mice. The trend becomes even more significant and pronounced when we take all 4 limb strength effects into the measurement. As we look into the underlying molecular pathways by which this treatment is having its effect, one of the ways that stands out is a reversal in the muscle fiber types and their metabolic pathways they utilize. One of the hallmarks of myotonic dystrophy and a few other diseases is a transition from having fast twitch fibers to having more slow twitch fibers. And we see that treatment with JUV161 reverses this trend, here particularly highlighted by the increase in type 2B fibers, shown in the far right bar graphs, and in the green highlighted fibers, and this is a histology section of muscle on the bottom for JUV161 treated mouse muscle. Treatment with JUV161 also increases the average size of the muscle fibers. here shown as a distribution of the cross-sectional area of muscle fibers in JUV161 in red against the vehicle-treated control group in blue behind them. And you can see there's a nice population in the middle there where you have an increase in those nice middle-sized muscle fibers following chronic treatment. We've also tested JUV161 in a number of other models that capture aspects of muscle wasting and atrophy, and we see broad therapeutic potential for ameliorating particularly the muscle strength and function symptoms of this disease that give us conviction for its clinical translation in myotonic dystrophy. Importantly, we also tested the molecule in human myotonic dystrophy afflicted precursor cells. In this particular instance, I'm showing some data collected by the team that shows that it strongly increases the expression of key markers of the progression from early muscle precursor cells into mature muscle fibers. Measured phenotypically, we also see that treatment improves muscle survival in the graph on the left, following JUV161 treatment here in blue compared to vehicle in red. And importantly, the fraction of those cells that form new muscle fibers in the graph on the right, and in the micrograph, those are the large green cells. Looking more globally at the effect of JUV161 treatment, we see that it has pronounced effect on the expression profile of human muscle precursor cells with the myotonic dystrophy mutation to restore the expression of components of muscle contraction and extracellular matrix that are important in the establishment and maintenance of muscle fiber integrity, as well as components of energy homeostasis that would allow it to utilize the metabolic components in the blood for generating the energy needed for muscle force. So in summary, I just wanted to highlight some of the work my team has been doing to develop JUV161 as a treatment for myotonic dystrophy, taking a somewhat different approach, as highlighted by Dr. Berglund, that can help treat important aspects of myotonic dystrophy being developed as toward a subcutaneously administered therapeutic with broad tissue availability. In our initial testing to date, we see good preclinical safety profiles that give us conviction about continuing our translation into the clinic, ideally, as I mentioned, early next year. As we work toward designing what those clinical trials could look like, we welcome input from the community and are grateful for the many stories we've heard so far and hope to hear many more. And toward that, please come find us at our poster tomorrow, where I and my colleague, Dr. Tabrizi, will be happy to answer any of your questions about the science and would welcome any stories about your journey you might be willing to share with us. With that, I'll thank the organizers, and please reach out to us on the linked website or again, in person as you find us. So thank you. I appreciate it.
Good afternoon, everyone. My name is Belle Gallion. I'm the Global Clinical Lead for the DM1 Gene Therapy Program at Sanofi. I am so honored and happy to be here with you today to share an update on our gene therapy program. Sanofi has had a long history of delivering therapy for rare diseases. So this has been going on for about 40 years now. And this commitment to the rare disease community is again reflected in our ongoing efforts to provide and investigate therapies for the DM1 community. We are here to listen to you and the DM1 community to hear how we can incorporate your thoughts, your perspectives, and insights into our clinical program. Before I tell you about our therapy, let us first review some basic gene therapy concepts. The twisted ladder that is shown in this slide is what is known as the double helix of the DNA molecule. Each strand of the DNA helix is made up of individual pieces called nucleotides. DNA functions like a manual, much like your TV manual that is made up of information and instructions needed to make proteins that are important for an organism to live, grow, and develop and function independently. This information is organized into different blocks called genes, which are passed from parents to children. Information from genes are transferred to an intermediate molecule called RNA before proceeding to DNA to protein production. One type of RNA is the messenger RNA, which transfers the information encoded in the genes from the nucleus to the cytoplasm, so the cell can then start producing protein. In DM1, as we have heard, and as you are all familiar with, the presence of these repeating CTG repeats creates an abnormally long DMPK gene, which results in long mRNAs that function like sticky traps that hold on to important proteins and prevent them from functioning properly, leading to the typical symptoms that we see in DM1. But what is gene therapy? Gene therapy delivers genetic material to change how a protein is produced by the cell. This genetic material is sent to a cell using a vector, much like your Amazon delivery person, for example. A vector delivers a package with specific message for the cells. The genetic material is delivered by infusion or injection, and it's then called in vivo gene therapy. We at Sanofi are investigating an AAV-mediated delivery of an artificial microRNA or a specially designed microRNA that binds and degrades the affected DMPK mRNA. So what you see in this slide is a typical clinical development process wherein discovery of a drug or a potential drug happens at the lab and is then tested in animals such as mice and monkeys in order to see if they are safe and effective. Once that is demonstrated, the typical process then progresses into looking if the same efficacy and safety is demonstrated in humans. So at Sanofi, we have developed a viral-based delivery system that aims to deliver a specifically designed type of RNA called microRNA, which aims to lower the affected DMPK mRNA level in cells. This, in turn, is expected to restore normal protein function. We have since then been able to successfully demonstrate lower levels of the abnormally long DMPK mRNA in animal models, and previously presented our results at the American Society of Cell and Gene Therapy conference this year. We are hoping to see similar results in humans and are currently actively developing our clinical program in order to find various ways that we can demonstrate this as effectively and safely in humans and within the community. We are hoping to start our trials within the next couple of months, and we are working very hard to move this potential treatment forward so it can be available to the community at the soonest possible time. As always, we are grateful to the MDF for giving us this opportunity to share this information and engage with all of you today. We are looking forward to further collaborations with you. Thank you so much.
Good afternoon. I'm very pleased to be there today. I'm Frédéric Legros, CEO of Artex. Artex is developing a lipid-conjugated anti-MIR to treat myotonic dystrophy type 1. I will elaborate a little bit more on the science and the development stage where we are today. So, who is Artex? Who are we? We are a Spanish company. We are located in Valencia, south of Spain. The company has been created in 2019 from the research led by Ruben Artero and Beatrice Lamuzzi, our co-founder, scientific co-founder. They are working on the modulation of MBNL protein in DM1 for a long time, and they discover that there is a specific microRNA named MIR23B that is regulating the expression of MBNL protein. Our objective now is to modulate this microRNA in order to increase the expression of the MBNL protein that is trapped. And many were describing the molecular cause of the disease. And so by upregulating MBNL protein, we hope to correct the disease. So we are developing drugs for disease with unmet medical need in which microRNA play an important role. Our lead compound is ATX01. It's an oligonucleotide targeting miR23B, linked to an hydrophobic conjugate in order to improve the uptake in muscle, and we get ATX01. So what do we want to achieve? As it has been already well described, the MBNL protein are trapped by the toxic mRNA of DMPK. What we discover at Artex and in the lab of our scientific co-founder, in addition to the trapping by the toxic mRNA, there is an overexpression of a miR named miR23B. These microRNA are there to regulate the expression of different type of protein, and miR23B is regulating the expression of MBNL protein. So in a way, you have a dual effect. You have first the trapping of the MBNL protein by the toxic mRNA, but at the same time, we discover that in patient, you have an overexpression of miR23B. So you have a dual effect. You have trapping of the protein, but at the same time, a downregulation and a down expression of MBNL protein. And this non-available protein leads to an aberrant splicing. So we want to correct that phenomena. So we developed this anti-miR, and you see on the left-hand side, this protein, ATX01, is linking miR23B. By doing that, we prevent the effect of miR23B. We get much more protein expressed. And at the same time, we also discover that by downregulating miR23B, we can impact the stability of the toxic mRNA, DMPK toxic mRNA. You see on this slide, on the top, we are testing the molecule in human DM1 myotube. You can see that we can increase MBNL protein by threefold by applying the drug ATX01. And at the same time, we downregulate the expression or the stability of the mRNA DMPK. We test that molecule in different models, mice. I will share you some data later on, but also in primate to evaluate the safety of the product. And at the same time, we were able to demonstrate that in primate, on the right-hand side, you can see that we can overexpress MBNL protein by applying ATX01 and also destabilizing the mRNA of DMPK. So I'm insisting on the fact that we have a dual effect and we are playing on the both sides. We test the efficacy of this drug in mice. We use the HSLR model. So you inject the molecule three times at day 0, day 14, day 28, and you evaluate the muscle strength and different parameters. You can see on the left-hand side, first of all, the functional rescue. So you see we measure the muscle force, and you see that by applying the drug at 6 mg, 9 mg, or 12 mg per kilo, you can significantly improve the muscle force in this model compared to placebo. We were able to evaluate the splicing. So you know that you have this aberrant splicing due to the trapping of the protein. And by overexpressing the protein, we can correct this aberrant splicing. And we have target engagement. So we have the drug working in different tissue. And here you can see the muscle. We have a dose-dependent effect. The more we inject the drug, the more we get protein expression, and the more we can correct the aberrant splicing. And we have the same dose-dependent effect in heart, in diaphragm, and we don't see a major uptake in liver and kidney, but we have also an uptake in brain. So we have a target engagement in brain, which is very important considering the cognitive defect. So we are at a stage of launching our trial. We hope to get first patient enrolled early 2024. We test that drug in different species to get the full package, to be able to submit that package to the European authority and US authority. We plan to run our trial in US, in Canada, and in Europe. The objective of this trial is a phase 1/2, so we go directly to patient. The primary objective will be to test the safety and the tolerability of a single and multiple ascending dose of ATX01. We will measure whether we get target engagement using biopsy. So we will evaluate the level of protein overexpress, MBNL protein overexpress in muscle, and the splicing index, which is very important to evaluate the effect of the drug. And finally, we will evaluate clinical endpoint, including measurement of muscle function, but also extramuscular function and patient-reported outcome. If you want to just get 5 key ways, take-home message, we are addressing a new target, miR-23B. We have a dual effect. We are playing both on the MBNL protein, and the toxic DMPK. miR-23B is well conserved from mice, rat, mini pig, non-human primate, and human. There are publications demonstrating that the modulation of miR-23B in animal model is safe. We have evidence of target engagements beyond the muscle, heart, and CNS. And finally, this drug is an oligonucleotide. There are many drugs like this one on the market. So we know how to produce this type of molecule. We know that we can supply product and manufacture that product for patient. So thanks a lot for your attention. I'm very pleased to be there and answer any question you may have. Thank you so much.
So I just want to thank our 4 great representatives from industry and remind you tomorrow we have 5 more talks, and I hope to see all of you tomorrow for those 5. Thanks, everyone.